Welcome to another lecture by MedicoMedics, Learning Made Easy, Microbiology and Infectious Diseases, Chapter 4, Virology, the Study of Viruses. So in this lecture, we will present what are viruses, talk about its structure, classification, the viral replication cycles, pathogenesis of viral infections, host defenses against viruses, what are some medically important viruses. Further on, we will talk about some antiviral drugs, some vaccines, and end with a summary. Now, viruses are these tiny infectious agents made of genetic material, DNA or RNA, and they are encased in a protein coat. Some key features of viruses include that they cannot replicate without a host cell, and they are much smaller than bacteria. Examples of viral diseases are the flu, HIV, COVID-19, etc. Now, what is the structure of a virus? So the basic components include a capsid. This is the protein shell that protects the genetic material. We have nucleic acid, so DNA or RNA containing viral genes. And then we may have an envelope. This is a lipid layer with spikes for host attachment. Now, viruses come in different shapes. So we have helical, like the rabies virus, icosahedral, like the polio virus, and complex shapes like bacteriophages. So viruses are like tiny packages. The genetic material is the instruction manual, right? The genetic material contained is the instruction manual, and the capsid is the box protecting it. Now, some viruses may add this extra layer, the envelope, which has these spikes to help it essentially attach to the host, to go into the host cell. And here you have an illustration. So these are the spikes, which it may or may not have. It has either DNA or RNA inside. We have the capsid protecting it, and then the envelope itself. Now, how do we classify viruses? We do it based on genetic material. Is it a RNA virus or a DNA? Is it enveloped or non-enveloped? What is its host specificity? Animals, plants, or bacteria? Now, examples can be RNA virus like influenza, DNA virus like herpesvirus. Then we have retrovirus like HIV. So classifying viruses is like sorting packages by their type and contents. Some carry DNA, others RNA. And some even come with tools to actually rewrite their host's DNA, like HIV. Now let's talk about the viral replication cycles. And we will use as an example HIV. So the steps here are important and high yield to remember. What happens first is attachment. So the virus binds to the host cell. Then we have entry. So the virus injects its genetic material or enters entirely. After that, we have replication. So viral genes take over the host cell machinery to make new viral components. Then we have assembly. So new viruses are assembled inside the cell. And finally, release. So the host cell bursts, also called lysis, or releases viruses gradually. And what is important here is that antiviral drugs often target specific steps. That's why you need to remember all of these steps. So here in this illustration, we have the HIV virus here. And first, it's binding, right? And then it goes through entry, fuses in. The virus capsid, through something called reverse transcription initiation, continues on to get to the nucleus of the host cell. And here, uncoding occurs, whereby the viral DNA has now successfully been delivered into the host cell. After that, it integrates with the host cell genetic material and then starts producing viral mRNA, continuing on to produce whatever it's producing and then exiting through assembly and release. Now, we will, in our lectures on HIV specifically, dive into greater details, talking about each of these steps in relationship to the HIV virus itself. However, here, what is high yield are remembering these steps, remembering that the virus will first bind, then it will enter or deliver its material. Then that material will go through replication and essentially taking over the host cell. Following from that, it will start producing whatever it is encoding or coded to produce or programmed to produce. And then it will start leaving the cell to go spread, causing damage. And another way to think about it is that viruses are like burglars. So they break into the host cell, use the resources to copy themselves, and then they leave. And often when they leave, they damage or destroy the host cell in the process. Now, what is the pathogenesis of viral infections? So, how viruses may cause disease, either killing or damaging host cells. This is called cytopathic effects. Another way is triggering excessive immune responses, so inflammation. And even though inflammation is fundamentally a good thing, if it's too prolonged, too excessive, it may actually start causing damage. And we have latency among viruses. So we have dormant viruses that reactivate later, like, for example, herpes. So you get it, and then it's quiet for a long time, and then it activates. Some examples of how these viruses may cause disease. So HIV destroys our immune cells. And if you don't have your immune cells, you will be immunodeficient. And when that happens, other pathogens that exist that would have normally been repelled by your immune system now attack your body and eventually overwhelming your system, even leading to death. And another example is influenza that damages our respiratory cells. Now, naturally, throughout evolution, our body has developed different ways to combat and defend against bacteria, against viruses, etc. So we have our innate immunity helping us against viruses. The first line of defense, for example, interferons and natural killer cells. When these fail, we have our adaptive immunity, whereby we have our antibodies that bind and neutralize viruses. And then we have T cells that help destroy infected cells. We use vaccines, of course, to stimulate immunity to prevent infection. And we go into depth in our classes on immunology, talking greatly in detail about all of these different cells. But for now, it's important to keep in mind that, indeed, vaccines are used to stimulate our immunity. We have an adaptive immune system helping us to fight off viruses. We even have our innate immunity trying to do that. When it fails, we have treatment, antivirals to do that. So let's mention just a few medically important viruses. We have the DNA viruses like herpesvirus, causing cold sores and shingles. Hepatitis type B virus, leading to liver infection. We have RNA viruses like the influenza virus and COVID-19 or SARS-2 virus. We also have retroviruses like HIV, causing, for example, AIDS. Now, just to put it into some clinical context to make it more memorable, let's dive a little bit deeper into HIV and how it causes AIDS. So, what happens is that HIV, all right, it enters the body and it targets our CD4 plus. These are actually called T helper cells, but these are part of our T cells. Remember, we mentioned we have our T cells and we have B cells. And one of these T cells is the CD4, also called the T helper cell. Now, HIV specifically targets this CD4 cell. And let me just mention that this cell is incredibly vital and important in our adaptive immune system, or in our immune system in general. So what happens is that HIV binds to it, and it binds to receptors on this CD4 and co-receptors on the cell surface. So the CD4 expresses, for example, let's see if my memory serves me right here, but we have CCR5, CXCR4. Now, these are co-receptors on this T helper cell. So the HIV binds to these. And when it does that, it then fuses with the cell membrane and releases, right? It releases its RNA into the host cell. So we talked about the first part was attachment and then entry, right? So it fuses and releases its RNA into the host cell. Now, what happens then? So the integrated DNA, viral DNA, now uses the host cell's machinery to produce new viral RNA and proteins. And these components then assemble into new HIV particles. And what happens then is that as HIV then continues to replicate, it will damage and destroy infected CD4 T cells. And furthermore, our immune system also attacks infected cells. So further destruction of CD4 T cell happens. So not only is the virus itself causing this damage, but our already existent immune system is identifying infected cells and it starts attacking it as well. So your immune system is now under great siege. Now, over time, what happens is that the progressive loss of CD4 will weaken the immune system overall, because I mentioned it's a very important immune cell involved in the totality of your adaptive immune system in general, important specifically for signaling, telling your immune system where to go and what to do, etc., etc. We are going into detail in our course in immunology, but for now, take my word for it, this is an important cell. So what happens is that as CD4 counts go down violently, the body then becomes unable to fight off opportunistic infections. And what then happens is that we have the development of AIDS. So AIDS is diagnosed, actually, right? AIDS is diagnosed when CD4 T cells drop below a certain number. And I will not mention this number because it's not relevant right now. But when our CD4 cells drop below a certain level, or when specific opportunistic infections or conditions escalate, right? And so what happens is that without treatment, this will lead to severe immune suppression and indeed life-threatening complications. So very opportunistic pathogens or causes of infection that would have normally been repelled if you had a normal CD4 count. You would have thrown these away, let's say. All of these all of these pathogens now become opportunistic. So they will seize the moment. So let's mention briefly some antiviral drugs. So what is the mechanism of action in general here? Well, they can block virus entry. Remember the steps again. We first had attachment and entry. So a mechanism of action of some antiviral drugs would be to block this entry. Like, for example, fusion inhibitors for HIV. We can use drugs to inhibit replication. So remember, when it's entered the host cell and delivered its content, now it wants to replicate. So we give a drug to stop this. Furthermore, after replication and all of that, what does the virus do? It begins to move towards releasing its product to go cause more mayhem. So we have drugs to block that, preventing virus release. Now, the challenge is that viruses mutate quickly, which leads to drug resistance. So research continues to find and try to stay a step ahead, but it's difficult because these are quite clever. But this is all in all what you need to remember. So you need to repeat and revise the steps of the viral replication cycle, and then understand, first of all, what these different cycles mean and what is happening during these different stages, but also then understand how our medical interventions actually work here. We stop here, or we stop it here, or we stop it here, etc., etc. Now let's talk about viral vaccines. So we have weakened viruses used to stimulate our immune system, to train it essentially, like the MMR vaccine, the mumps, measles, and rubella vaccine. We can use killed virus, like in polio vaccine. We have mRNA vaccines, like the COVID-19 vaccine. And what are some benefits? Well, preventing infection and reducing disease severity. And another challenge here would be that mutating viruses may require updated vaccines, for example, the flu. So vaccines are like training exercises for our immune system. We are presenting it with a little part of this pathogen, enough to stimulate our immune system to start building protection and antibodies, essentially, cells, so that in the future, whenever this thing appears again, we already have a protection against it made in our system. But not enough is given to stimulate disease. Now, this isn't always bulletproof, but that's the general idea here. Viruses and biotech. So, for example, in gene therapy, viruses are used as vectors to deliver therapeutic genes. In vaccine development, we're using viral platforms. In cancer therapy, oncolytic viruses are used to kill cancer cells. So scientists can use viruses for good by turning them into delivery vehicles for medicines, vaccines, or even cancer treatments. So essentially, it's like using a hijacked enemy tool to help people. So that's one way to stay ahead of the game. But viruses can be very clever and even recognize what we are trying to do sometimes, so they find new ways to adapt and create further problems. And this is why all of this is very interesting. So in summary then, viruses are unique. They are not alive, but they are capable of causing diseases. This is very philosophical. And understanding their structures and life cycle indeed helps us fight infections. Advances in virology are improving treatments, vaccines, and biotech. So viruses are strange and fascinating. Indeed, they are not alive, but they can wreak havoc or even be used for good. So by studying them, we can protect ourselves and also unlock new medical advancements. And that's the end of this lecture. Thank you for listening. Continue now to Chapter 5.